GI system has already been covered by you in physiology, and you know the structure of the small intestine, stomach, that the viscera, they do contain smooth muscle. Clear? Smooth means there are no striations. In skeletal muscle, there are transverse striations, but in smooth muscle, it is not so. It contains actin and myosin too. এইদিকে কেউ নাই কেন? হ্যাঁ, সব একদম ওইখানে যাচ্ছে। সেল অ্যাডিশন মলিকুলস, হ্যাঁ, এই দিকটা কেউ বসো, এই দিকটা, এই পাশটা। No transverse striations, and in the anatomy dissection hall, you also handled these structures. I think your abdomen has been covered. The structures present in the organs present in the within the abdomen, you have seen. Hollow structures, they contain in their wall smooth muscle, and so it is called visceral muscles. And they are of two types mainly. One is visceral, and the another, and they are the abundant, and other is multi-unit. Multi-unit. They are present in few situations. Smooth muscles, in general, they have their structure spindle shaped. They are spindle shaped, and they are in every viscera. The character of the smooth muscle is different, and it is specialized for that organ. But there are gross similarities between them. Whether it is multi-unit or single unit, or unitary or visceral, they are formed of individual cells of this type. They contain a nucleus, and in this way they are disposed. In layers they are disposed. Yeah. In layers. Cross section of this small intestine. Outer is the peritoneal coat or serous coat, and then there is the longitudinal muscle coat, then circular muscle coat, and then there is the muscularis mucosae, and then the mucus coat, and at the lumen, at the epithelial cells. This is the structure. And visceral smooth muscle, they are disposed in this way, and they have physical contact with them, and they also have these nexuses or gap junctions between them, and they contain actin and myosin too, like skeletal muscle, of different isoforms, and their myosin content is relatively much less in comparison to that of the skeletal muscle. and there is sliding between the actin and myosin filaments that is responsible for the contraction. But there are no striations in that sense. Striated muscle is present within the heart and in this skeletal muscle, and they are disposed in that fashion. But here they are disposed in another way. At their membranes there are dense bodies. Dense bodies. From the dense bodies there is the actin filaments. These are the actin filaments. By means of actinin they are, and within this, and there is much smaller number, there is the myosin filament. These are the actin filament, these are the dense bodies. Dense bodies. Dense bodies. This is the actin filament and this is the myosin filament. The intermediate is the myosin filament, and there are interdigitation. And these dense bodies are present within the cells and also close to one another within the near this membrane, dense bodies, and from which there is the actin filaments. And this contraction, so there is a group of muscle can contract. Group of muscle can contract. Individual cells, suppose individual cells, the diameter is 10 to 20 μm and their length is approximately 200 to 250 μm. It is variable. In this way they are disposed in layers. In the small intestine there are this outer is the longitudinal layer, inner is the circular layer, and they are formed of this, and there are gap junctions in between them, so that the action potential can propagate easily from this side. And in the multi-unit smooth muscle, they are separate. There are no gap junctions between them. Suppose the iris, muscles of the iris, ciliary body, and some other part of the, that is erector pylorium muscles, that is responsible for the movement of the hair of the skin, and they are smooth muscle and they are multi-unit variety, and in that case the cells are separated. These cells are separated and they are not connected to each other by means of these gap junctions. And surrounding this there is a thin connective tissue coat with glycoprotein, basement membrane like structures, but that is also applicable in this case also. And there are no gap junctions, and they have separate innervations, multi-unit muscle. And this innervation of these muscles are different from that of the skeletal muscle, and it is called synapse en passant type of synapse en passant. Suppose synapse en passant means that whenever a postganglionic neuron that is, then it is branching, branches are going, and there are some varicosities at some distances, varicosities, and these varicosities, they contain this neurotransmitter or etc. And when in this process it is going, it is supplying, suppose one cell, and it is supplies the other cells, in this way. And there are no specialized connections like neuromuscular junction of skeletal muscle, varicosities, means swellings, and that contain these neurotransmitters. And in case of multi-unit smooth muscles, they are separate and they have, suppose they have their individual innervation, similar to that of the skeletal muscle. Clear? In skeletal muscle, the cells are all isolated. There are no gap junctions between the skeletal muscle cells. And in the multi-unit type, it is also the same type. Suppose it is the nucleus. But in case of more abundant visceral smooth muscle, that is forming the walls of the viscera, from the lower part of the esophagus up to the anal canal, they are present. In the urinary bladder, uterus, ureter, and also blood vessel, muscles of these blood vessels, they are all visceral muscles. Multi-smooth muscle, the distribution are few sites, and their requirement is that they cannot be controlled by us. Suppose the constriction of the pupil. Pupil allows light to enter, and what is the adequate diameter produced, we cannot control it, but it is required. Whenever their functions are beyond our control, but fine adjustment is required, and then the nature is given this duty to the smooth muscle, multi-unit smooth muscle. Suppose in case of ciliary muscle, the diameter of that is the thickness of the lens will change, and it must be accurate, but we have no power to control it by voluntary action. And for this reason, nature has given this duty to the multi-unit smooth muscle. Here. Or where there is no requirement of voluntary activity, the body should go with its own purpose, and there the nature has given responsibility to the smooth muscle, visceral smooth muscle. Suppose we are eating, only the upper part of the GI tract you can control before swallowing. After it goes into the stomach and small intestine, we have no control, we require no control, but it is controlled by the autonomic nervous system and the smooth muscle. They do whatever that is required for us. Movement, secretion, help in the secretion, in the reabsorption, everything is done with the autonomic system. Similarly, in case of, suppose what should be the distance? In case of accommodation, we try to accommodate, and it is controlled by the smooth muscle but not by the voluntary muscle. Yeah. And it is visceral type of smooth muscle, they are in sheets and they are connected by means of these gap junctions. So the impulse can spread, and all the cells does not need this type of synapse in patient. It is going. In case of skeletal muscle, you can remember that when the neuron, the action of the anterior horn cell come to the muscle, near the muscle, and it divides. And a group of muscles, skeletal muscles supplied. The individual action and the group of these muscles, they form the motor unit. But here it is different in that case, and a impulse is going through, it supplies this and supplies this in this synapse in patient, in both in sympathetic and parasympathetic division. All the same thing is going on, and not all the cells does not require innervation, and it will spread from this cell via the gap junction and via the gap junctions in this way. So they will spread. So they behave as a syncytial mass, like a cardiac muscle. Yeah. Structure of actinin and myosin 2 is apparently similar, but there are different isoforms, and there are tropomyosin, but troponin is absent. Troponin is absent. And in that case, this excitation contraction coupling is to some extent different from that of the skeletal muscle. In case of skeletal muscle, the excitation and contraction coupling is produced by which mechanism? That is, the excitation is responsible for contraction in skeletal muscle. এটা তোমাদের জানা আছে কারু? হ্যাঁ, মনে আছে এক্সাইটেশন কন্ট্রাকশন কাপলিং স্কেলিটাল মাসেলের ক্ষেত্রে কিভাবে হয়? নিশ্চয়ই জানা আছে তোমাদের, জানা নেই? এটা পরীক্ষা, এটা তো দেওয়া হয়েছিল, এটা জানতে হবে তোমাদের। বাই ট্রোপোনিন সিস্টেম। Troponin has 3 subunits: troponin I, troponin T, and troponin C. And when calcium ion unites with the troponin C, then there is some changes, so that this is the myosin head can unite with the appropriate site of the actin, and then myosin ATPase activity will increase. There is liberation of ADP and PI and energy, and there is power stroke, and there is movement of the actin filament of the myosin. But here is troponin is absent. So this function is done by something else, and it is done by calmodulin. Calmodulin. So a structure similar to that of the troponin C, calmodulin. It is a cytosolic protein. It can bind with 4 calcium ions, and the structure is extensively conserved. Even in the plant kingdom, there are similar structures, and they can bind with 4 calcium ions, and their number is 148. And at 1 and 5 there is trimethylated lysine group, and that is preserved. And when calcium binds with it, they form calcium calmodulin complex, and it then causes the activation of some kinases, myosin light chain kinases. Myosin light chain kinases. Suppose N terminal, C terminal, it is the formula of, that is the myosin 2, similar to that of the skeletal muscle, and they are called these heavy chains, and they are called, they are, and 1, 2, 3, 4 they are called এগুলো কিছু কিছু স্মরণে রাখতে হবে, কিছুটা পড়তে হবে, এগুলো নিশ্চয়ই পড়া হয়েছে। হ্যাঁ, এগুলো হচ্ছে লাইট চেন, 4টে লাইট চেন আর 2টো হেভি চেন। এটা নিয়ে হচ্ছে তোমার একটা হেটেরোহেক্সামার তৈরি হচ্ছে, বোঝা গেল? হেটেরোহেক্সামার। এখন এর মধ্যে 2টো যে লাইট চেন আছে, 2 লাইট চেনস, আর phosphorylatable light chain, and other 2 are regulatory light chains. And this, in this smooth muscle, this myosin light chain kinase is activated by this calcium calmodulin complex somehow, and it causes activation of myosin light chain kinase, and there is phosphorylation of myosin light chain. Suppose it is the phosphorylation of the myosin light chain. And when this occurs, then there is increase in the myosin ATPase activity, and there is binding of the actin and myosin filaments. Actins are much more numerous, Actins are much more numerous in this smooth muscle than in the skeletal muscle. In spite of that, there is contraction. And this contraction is such that the lumen may be obliterated almost completely. There are some sphincters, smooth muscle sphincters within the GI tract, pyloric sphincter, and within the blood vessels. Whenever there is vasoconstriction, there is occlusion of the lumen, and it is required. Suppose whenever there is some injury, then the system tries to stop the bleeding completely. For complete stoppage, there must be almost complete constriction, so that the lumen is obliterated. Further bleeding is stopped, and it is a common experience. Small cuts we are receiving in everyday activities. We do not care, and they become blocked. Initially by the platelets, and also by the contraction of the smooth muscle that is present. At the level of the arterioles, there are the arterioles and smooth muscles, and they can contract, and they can close the lumen. In case of skeletal muscle, this type of complete contraction is not possible. The shortening is limited. Shortening is limited. Shortening is limited. 1/3 of it can contract up to 1/3 of its length. Suppose it is the biceps muscle or flexor muscle group, you can contract maximally up to, that is the 2/3 of the initial length. That is up to, so 1/3 of this length you can contract, not more. But in this case, it is possible that more than 80% of this length can be reduced whenever required. Otherwise, how it is possible to constrict it completely? Suppose in this case, in a case where suppose it is the lumen, it is the contractile, it is the muscle, and whenever there is requirement, it can close it so that it can almost completely obliterate this muscle. Obliterate the lumen. It is possible, and it is going on within the body. Yeah, it does. So it is the function of the smooth muscle is to some extent different, and different of in their structures. Side polar arrangement is possible for that. In case of skeletal muscle, you have seen suppose it is the you can remember it that in this way they are disposed in overlapping fashion, and their polarity is different on either side. In the center is the M line, you can remember, skeletal muscle, and it can bind with the actin, and it can move in this way, and contraction is possible. But in case of smooth muscle, this type of movement is also there, but there are also side polar arrangement of side, that is on one side. Suppose on one side and the other side, all through it is opposite side. opposite side, opposite side. So that side polar everywhere, so it will draw the actin in this way, it will draw the actin in this way. So that there is, this lumen can be obliterated, almost obliterated, side polar arrangement. On one side in this direction, if you take in this way, if you take in this way, suppose is the myosin filament, one side on this direction and other is on this direction. So it will move in this way and it will move in the opposite way. Side polar arrangement. On one side this pattern and another side this pattern. So that then there is overlapping, much overlapping, and the lumen may be oblate. Side polar arrangement. It is called side polar. Myosin filament on one side in this pattern and another side opposite pattern. And this movement is in the opposite side, opposite side. So actin filament on one side, on one direction and other side in the opposite direction. তাহলে সেটা এরকম ভাবে ইন্টারডিজিটেট করতে পারবে, বুঝা গেল? যেমন আমি যদি এরকম ভাবে যদি করি, একদিকে এই যে মাঝখানে মনে করি মাউসের মলিকুল আছে, মাউসের মলিকুল আছে, একটা আবার হাতটা একটা এদিকে যাচ্ছে আরেকটা ওদিকে যাচ্ছে, তারপরে সেক্ষেত্রে আমার ওভারল্যাপিংটা অনেক বেটার হচ্ছে। এই জন্য 80% অফ দি লেংথ সেখানে কমে যেতে পারে। So that the lumen can almost obliterate. Now this electrical activity of the smooth muscle, we also have to know electrical activity. And here is there is also excitation contraction coupling in most cases, except some condition in multi-unit smooth muscle. In visceral smooth muscle, there must be excitation in the form of depolarization or even action potential generation. Action potential generation. And when suppose a part of this smooth muscle containing viscera, if this experimental animal is sacrificed and a duodenum and upper part of the jejunum is placed in appropriate fluid that contains the electrolyte and air in suppletion to provide oxygen, and it can contract outside the body. So that in this case their contraction is self-generated. They are not generated by the nerve impulses. In case of skeletal muscle, all the impulse coming from the nerves, if the nerve is damaged, then there is the muscle becomes paralyzed. Lower motor neuron type of paralysis or upper motor neuron type of paralysis. But in case of smooth muscle, if it is taken out of the body in different parts, 2 centimeter, 3 centimeter part, and kept in appropriate fluid containing this nutrition, electrolytes and air or oxygen in suppletion, it can contract. You can see with your unaided eye that they are contracting. So then they are supplied by the nerves, autonomic system, but they are contraction originates within itself, and they are always active, almost always active. Irregular contraction is going on, so that there is also some changes in the electrical activity. But whenever there is a relatively quiescent condition, and in that case the membrane potential has an average value of minus 50 millivolt, minus 50 millivolt. And they may be sinusoidal type or they may be of different types, like pacemaker type of potential. Pacemaker type of potential is present within the SA node, AV node, normally SA node. pacemaker potential. And you have, I think you have all learned about how to draw this pacemaker potential. Then this potential you can draw in this way. In this way, this type of potential can be seen in this smooth muscle. They are slow process, but similar to that of the pacemaker potential in cardiac muscle. Cardiac muscle. Suppose it is the resting membrane potential, relative minus 50 millivolt. Suppose it is minus 65 or so, and in that case, excuse me, minus 45 or so. 40 millivolt, and then there is the action potential, and then it is the zero level. It may reach or may not reach the zero level, and it is the repolarization, and from this level, then with parallel to it, you draw a line, and parallel to this in this line, and parallel to this line, so that in that way you can draw this the pacemaker type of potential. Yeah. It is slower than in cardiac muscle, but in this way you can draw it in this, or it may be of sine wave-like fluctuation. In this three lines, three parallel lines, and you draw in this way. In this type. Suppose it is minus 50 millivolt, it is minus 55, it is minus 45 millivolt. And suppose at this peak, there is the action potential. Or in some cases, you will find that of the cardiac muscle, plateau phase, prolonged repolarization, plateau phase, working myocardial cells, and their action potential will be this type of variety. Plateau phase. Suppose it is minus 50 millivolt. In case of smooth muscle. Suppose it is a plus 5 or 0, and this is plateau phase. But it is similar shape of that of the cardiac muscle. Yeah. So all sorts of can be found, and it may be found in at the same time in a particular muscle also. Some cells showing this type of pacemaker type, it is sinusoidal type, and some also this type. It depends upon the behavior of this. The sodium channels, there is calcium channels, there is potassium channels, they will give to this type of variations. But commonly, this type of is common. Smooth muscle. And whenever this type of, suppose in case of small intestine, you are familiar with the type basal electrical rhythm, and migrating myoelectrical complex, MMC, and they are of this type, and whenever there is, and it may be at the peak, it may be in the ascending limb, and it may be in the descending limb also. Whenever, and it is the when it is the basal, when it is the basal type of potential change, that is, this potential is not fixed like that of the skeletal muscle and also the cardiac muscles, besides the pacemaker cells. They are always fluctuating. Due to entrance of calcium ion primarily or sodium ion, and they are always fluctuating. And at their peak, there may be this action potential generation. This action potential duration is much more. Duration is much more. And this excitation contraction coupling is a slow process. Slow process. In case of skeletal muscle, if you remember, then it is the muscle cell, it is minus 90 millivolt is the membrane potential, and then these are the features. And the spike potential, suppose it is 2 to 4 millisecond in this their duration. Skeletal muscle cells. In neurons it is further less, 1 to 2 millisecond. But in this case it is much slower process. Suppose it is 50 millisecond. In this case, if we consider this, it is 50 millisecond. From its value, 50 milliseconds. 50 milliseconds. Spike potential is 50 millisecond. And after 150 millisecond, then this contraction starts and may reach its peak after 500 milliseconds from this point. 500 milliseconds. 500 milliseconds. So it is a slow process due to this myosin ATPase activity is slow. বুঝা গেল? এর যে কুইক কন্ট্রাকশন এটা করা যাচ্ছে, রিল্যাক্সেশন করা যাচ্ছে, তার মানে এখানে আমাদের র্যাপিড সেই কন্ট্রাকশনটা সম্ভব। ক্রস ব্রিজ রিসাইক্লিং সেটা কুইকলি হচ্ছে বলেই তো এটা সম্ভব হচ্ছে, কিন্তু এদের ক্ষেত্রে সেটা হবে না। স্লো প্রসেস, এখানে কোনো তাড়া নেই। There is no urgency. Suppose after eating, you are sleeping after the meal, and for this reason it was not possible to continue some classes for you. Yeah. And you forget about the food, and it is in the stomach and small intestine, and then they are progressing slowly. There is no need of urgency, and there is peristalsis. Initially there is receptivity. When you are eating, there is relative relaxation of the stomach, but after that peristalsis starts, and there are some pacemaker cells. They are called interstitial cells of Kagel, and they are spider-like cells, stellate cells, and they will cell processes, and they stimulate the neighboring cells, and same cells at these gap junctions, and in this way they can propagate impulses. Suppose, and when you have read suppose it is the circular muscle and it is the longitudinal muscles. Proximal part of the intestine, the cells of Kagel is that part. In between this, there is some space, and there is mesenteric plexus, and it is the submucous plexus. And close to this is the cells of Kagel is close to, there is outside the circular muscle. very near this plexus, mesenteric plexus, and at the colon they are near this side, at the colon. It is the small intestine and it is the large intestine, colon, they are their disposition. Whatever may be, they are the pacemakers and they generate action potential automatically and they propagate, and some part is involved. Some part is involved. And not in the fasting state. Not in the fasting state. Whenever there is this movement, migratory myoelectric complex, MMC. But here, in other times, there are some activity, basal electrical rhythm, and their frequency is different in different part of the small intestine. In case of stomach, it is 4 per minute, and at the part of the ileum and at the jejunum or duodenum, it is 12 per minute. At the ileum, it is 8 per minute. 8 per minute. And then in the colon, it is initially cecum, it is 3 per minute, and it is much more when it goes to the sigmoid colon in the basal electrical rhythm. And where this type of there is this slow waves you can see, basal electrical rhythm, and they are not associated with contraction. They are not associated with contraction. A contraction occurs whenever there is generation of the action potential, and the number of action potential depends upon whether it is stimulated or not. Stimulated or not. Suppose in some cases parasympathetic stimulation is activated. And in that case, there is the number of action potential maybe more than this, and there is more force is generated, action potential. More force is generated. On the other hand, if there is sympathetic stimulation, then this action potential decreases. And normally there is a basal tone. We discussed that a muscle can contract individually. Basic contraction is not dependent exclusively on the nervous system, but it can regulate it. In different figures in your text, suppose in some cases intestinal preparation is a basal condition, it is this minus 50 millivolt roughly. Whenever there is depolarization in that sense, and by acetylcholine or so, and then there is increase in the tone and the action potentials do develop, like acetylcholine, cold, or even stretch, STRETCH. Stretch. Whenever there is stretch, it can contract. Some channels will open, calcium channels will open, and this calcium is responsible for contraction. And on the other hand, whenever there is sympathetic stimulation or epinephrine that is given in the media, and then there is diminution on the tone, and there are very few, there is this action potentials, like epinephrine, sympathetic stimulation. It becomes more negative, and it becomes less negative. Now this excitation contraction coupling we have to consider, and you can compare with that of the skeletal muscle. Suppose when you are taking the case of nerve stimulation, acetylcholine is added. Previously, this was included in the practicals. Then we have to sacrifice some white mouse or so, and their intestine is taken in thyroid solution, appropriate solution, and given air or oxygen in suppletion, and we can see the movement. And when a few drops of acetylcholine diluted, much diluted, 1 in 10 to the power 5 or so, drops is given, then we can see that it contracting with more force with the unaided eye, and you can record this in your mechanical event, you can record, and it is electrical record is given in the text to make you understand. So acetylcholine, what occurs, acetylcholine acting upon some muscle, smooth muscle, suppose intestinal smooth muscle, then this acetylcholine bind with the appropriate receptors. Suppose it is the cell membrane, and acetylcholine unites with these appropriate receptors, and in this case it is the G protein coupled receptors, and they are serpentine receptors, in this way. 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5, 6, 7 serpentine receptors. It is the N terminal, it is the C terminal. And third loop, 1, 2, 3. This loop can bind with this G protein, GTP binding protein, G protein. And then normally it is GDP. Normally it binds with the GDP, and when this acetylcholine binds it, then it is converted into G alpha, GTP, and alpha beta, they separate. GS within the membrane, and ultimately there is activation of the calcium channel, and there is increase in the calcium ion concentration, within the cytosol, G protein, alpha, beta, gamma, G alpha. Normally it is bind with GTP. When this activity is there somehow this GDP is changed into, replaced by GTP, and it will separate. And ultimately with this alpha GTP, GS alpha GTP unit, or this that causes ultimately the entry of the calcium ion. Then calcium ion will bind with calmodulin, calmodulin. And this complex activates then this myosin light chain kinase. And myosin light chain kinase causes phosphorylation of myosin light chain, phosphorylation. And there is activation of the myosin ATPase activity. Activation of, that is the myosin ATPase activity. It will increase, myosin ATPase. And then there is actin and myosin, myosin binding. And there is a contraction. And it is a slow process, and it is the same contraction. And after the contraction, then what occurs? There may be relaxation, and relaxation means that this phosphate must be removed. There is myosin light chain phosphatase, and they will remove this part, and then there is relaxation. Or there may not be action potential, or there not be, and in that case there not be action potential, but after that relation may not occur. There is no further action potential in spite of that potential change, and in spite of that there is contraction will continue for some time. It is called latch bridge mechanism, LAT, bridge mechanism. There will be contraction, but there is minimal change in the ATP consumption. So it is very much economic. And energy requirement of smooth muscle contraction is very low. And it is a slow process. Suppose it is this, if this is the speed of this muscle, and it is slow process, very slow process. But the force generation ultimately even more than this, even more than skeletal muscle. Force generation with minimum expenditure of the energy. Clear? Intestinal smooth muscle, they are essential muscles. We are taking food and they are taking care of, and all sorts of movements are possible by the smooth muscle. They are pushing the food, they are segmenting this, they are mixing the contents, they help in the absorption. In some cases, they have tonic contraction. Principal contraction is peristalsis, and then segmentation contraction. And in some cases, there are the tonic contraction. But their energy consumption is very poor. Even it may be as low as 1/300 of that of this produced by the tension of this skeletal muscle. Energy consumption is very low, but they are more effective. In case of skeletal muscle, per square centimeter, what is the tension strength produced? 3 to 4 kg. 3 to 4 kg per centimeter square. But in case of smooth muscle, it may exceed that. It may be 4 to kg, 6 kg per centimeter square. You can remember that the parturition is the function of the smooth muscle of the uterus, and they will discharge the newborn regularly. So whatever the force is required, if there is obstruction, and then they require some foreign hand, but nature is made is everything appropriate. Its force is sufficient to deliver the baby. Similarly, if there is a need, there is constriction of the blood vessels, contraction of the other part of the hollow organs, so that huge amount of force can be generated. If a stone obstructs the ureter, there is renal colic, and it is strong enough to push it outside. Normally, it can occur. Some of the gallstones may be expelled via the sphincter by the contraction of the bile ducts or gallbladder. So huge force can be generated. But expenditure of energy is much less than in comparison to that of the skeletal muscle. And contraction of the multi-unit smooth muscle, that is discrete, very discrete, fine and precise. You do not know what should be the thickness of the lens for accommodation, but the smooth muscle can do. They are acting like a computer, and visceral smooth muscle is responsible for our life, because if we can digest anything, then our life is not possible. They are doing everything at the expense of nothing, and they are like the trees and the plant life. They are giving us everything at almost expense of nothing. We are cutting trees, but they are giving us everything. Smooth muscle is comparable to that of the plant kingdom. They are giving us everything at the expense of nothing, and we are destroying. We are destroying our surroundings by cutting them and killing them. Yeah. So this is this, and in case of skeletal muscle, it is this troponin made here. Troponin is absent and take place by the calmodulin, calcium calmodulin complex, and it unites with the calcium, and this complex causes the activation of myosin light chain kinase, and light chain they become phospholipase. In skeletal muscle also, they may be phosphorylated, but it is not essential for their action. But in this case, they are essential for that, and that is the phosphatase enzyme. And in this case, the myosin light chain kinase, it can be phosphorylated or dephosphorylated. Dephosphorylated form is an active form, and phosphorylated form is an inactive form. And smooth muscle has one property that is different from the skeletal muscle. There is no length tension relationship, and this property is called plasticity. They behave like a viscous mass than a rigidly structured tissue. I think you have all gone through that part of this muscle length tension relationship, and there is a resting length. There is a resting length in which the developed tension is maximum. But in case of smooth muscle, visceral type of smooth muscle, it is not so. It is not so. And they are variable to length. If a smooth muscle is stressed, initially the tension increases, but it gradually decreases and even go below the original stress. It is a phenomenon called plasticity, and it can be easily demonstrated in case of urinary bladder. Urinary bladder. Bladder can be made empty. After empty, a catheter is introduced, and its pressure can be measured with introduction of small amount of, that is the fluid or air, and then there is a cystometrogram. It is important for the examination, not only for the smooth muscle understanding. Cystometrogram. It is the volume and it is the pressure. CMH2. Bladder made empty, and by means of a catheter, air or fluid is given, and there is a prolonged plateau, and then when this micturition reflex is produced, then there is abrupt increase in this pressure. Suppose it is 80 centimeter of water, 80 centimeter of water, it is 40, and it is 20, and it is divided into 1A, 1B, and 2. This classified in this way. It is up to 400 ml. 400 ml. When it is 150 ml, then we are become conscious of our fullness of the bladder. But when it approaches 400 ml, it is very difficult to control it, and micturition reflex is produced. And the pressure may go up abruptly at 80 centimeter of water or so. And it is due to this activity. Suppose in this position, there is initially increase in the pressure, but gradually it will die down to this level, and increase in this way and gradually die down to this. Within minutes it will come. And there is a long process. And it is also example of law of Laplace. You have read the law of Laplace in your cardiovascular system. P is equal to 2T by R. In a spherical viscous. In a spherical viscous. P is equal to tension, it will operate in this way, and pressure is in this way. And this is the radius. P is equal to 2T by R. If there is increased fluid, then pressure will increase, and if it is elastic, then this R will also increase, and it will remain same. And whenever there is much increase, and in that case the micturition reflex. If the micturition reflex is not there, it should enter in this way, and gradually it will go on. Yeah. It is an example of plasticity. It is also present in other part of this viscera, smooth muscle viscera. And there are some differences in the function or peculiarity of the function of vascular smooth muscle or so. Then you have to read yourself. Next day we will start with respiratory system. That is much more important, and it is more theoretical than in practical. Yeah.